then they release the similar different kind of wavelengths and which comes out in the form of color or different shades. So now if we see the coloring substances, several inorganic substances or synthetic substances have been developed and which have been successfully used as coloring substances, particularly for feed of animals, particularly in cosmetics and even in food items. And when we talk of synthetic substances that are being used in food systems, we have the issue of food safety. So for that, what we do? We have to basically undergo, we have to subject this developed substance, synthetic substance, to toxicological testing. So toxicological testing at different levels is performed. For example, we can go for toxicological testing at cellular level. We will go for cell culture and see if any kind of degradation or deformations occur in the cultured cells. Or we can then move on to the in vivo studies using animal models, wherein we can use different doses of the tested substance, be it a colorant, be it a flavoring substance or any such substance. So we establish safety that way, that there is no observable adverse effect in the animals or in the tissues that we are testing, and then we subject it to different doses. Obviously, at a higher dose, some adversity will come, but if that dose is significantly lower than the dose that we are intending to use, then we will say, okay, this is safe for food consumption, because if we are adding 5 ml of a colorant in 1 kg of a beverage, and we are testing that 5 ml is not a toxic dose to animals or some other testing species, then we establish its safety. But over a period of time, the colorants that were developed, they either couldn't qualify to be called as safe, or there were restrictions for its use, that it is to be used below this level, it is to be used only in this and this product. So there were restrictions in their use. And then with the advancement of studies, several colorants and synthetic ingredients these are even banned for food use. There's a long list of that when it comes to colorants and other things. So, but one beautiful thing with these synthetic colorants was that these were very stable. These were highly stable and were not affected by processing and storage conditions. So that was the good thing, and that was the attraction with these. But so the technological aspects are there, but at the same time we have our focus also on the health and well-being of the humans. So that is the basically genesis that we are looking for the alternatives. So natural colorants are there that are present in foods. We need not to basically give any colorant to tomato products. You will never see that any tomato product, be it puree, be it sauce, be it anything, even if it is very severely processed, thermally processed, but we need not to add any colorant to it, because despite that there may be degradation of lycopene, still it will retain sufficient quantities to attract our eyes. Now, so coming basically from synthetic towards the organic ones and natural ones, we have two options with us. Number one is that I will just make you things understand, then we can just run through the presentation. That is off late what I have been doing. We have two options with us to get away from these inorganic synthetic colorants. We can either extract it from the natural sources. We have foxconn, some of our students have worked on that. We can extract lycopene from tomato, these peels or tomato directly, and we have like marigold, we can extract carotenoid. So we have your product that is beetroot, red coloring pigment which is there. So we have certain natural substances which are very rich in coloring pigments, and if we will remove that, we will extract that, we can add those as colorants, or we can use their wastes. Usually we cannot, in terms of when we talk of economics, we cannot afford that separate way. Beetroot is used as a coloring agent, or it is used as a source for extraction of colors. Yes, if we have a beetroot juice, the pomace of that is still rich source of the pigment, we can, instead of throwing it away, we can use that. So there are n number of instances which we can use and which have been used, which we have used successfully. That we have extracted it from the natural sources, particularly from the plant sources. Like we have good examples in the form of lycopene, we have marigold, we have beetroot, and likewise. But one or two issues, main issues, are basically there with these pigments. One is general, that is with all the organic substances, that is the stability. Stability to processing as well as stability during storage. So during the processing, there is every apprehension that these pigments will fade away. These will be degraded into some other components, their sugar moiety may be lost, then some other functional group may be lost, and then these are subjected to several chemical changes, ultimately resulting in a substance that is no more coloring substance. And then during storage, again, on exposure to light, on exposure to oxygen, slow reactions may occur, which may again result in loss of color in these pigments. So that is one issue. And one more issue that is there is the yield. So normally, if we process 1 kg of carrot, not the pomace, the carrot, if we process, we will get around 70 mg of carotenoid only from it, beta carotene, that's a color substance. So again, yield is an issue. We need a huge and large raw material in order to make it workable on the commercial scale. So that is one main issue with this. And the third issue is that when it comes to the pigments from the natural sources, we don't have a single molecule there because we extract it with some solvent. So every substance that is solubilized in that solvent, that will come out. So there will be alpha carotene, there will be beta carotene, maybe dominance of one is there. But we cannot say that the color is because of a single molecule or a single compound. So a combination of compounds basically gets extracted that we have to use. We cannot then go for purification and then get a single compound. So and then the second alternative that that we have with us. So one was that we can use the natural materials or the agro wastes that are pigment for the extraction of the color. Then we can obviously see the encapsulation for part of it and stability part of it. That is a separate chapter. Then second option with us is the microbial colors. That is the title of the talk, that there are several microbial species which produce colors in their body. And mainly we have the members from algae, we have members from fungi, and we have members from bacteria. So these are the main microorganisms which produce colors. So what we do? We culture these microorganisms under the controlled conditions of lab or industry, what we call fermenters or bioreactors, give them the ideal conditions, both in terms of the environment, relative humidity, temperature, as well as in terms of the nutrition, that is the growth media, so that their growth is as fast as possible, and once they grow at a particular stage of their life cycle, they produce pigments within their bodies. Then we harvest these microorganisms. We harvested these microorganisms, that's called downstream processing. This is the culturing, then we have downstream processing, that is we harvested these cells of the microorganisms from either from their media, and then we disintegrate their bodies and remove the colors. And those colors then we can purify and we can use those as the coloring substances, be it in drug industry, be it in food industry or somewhere else. So there are several success stories that are attached to it. We have successfully developed biocolors or microbial organ colors, and these colors, if we see now with the basically advancement in their studies further, that these are not only the coloring substances, these have also health promoting properties. Say if we talk about beta carotene is primarily a pigment. It imparts color, that is from yellow to orange color. But at the same time, it has very high antioxidant activity. It has anti-cancerous properties as well. So, besides that, we are using these pigments as coloring substances. At the same time, we can claim that these are nutraceutical substances as well. A very good example is astaxanthin. Though beta carotene has a nutritional role because it is a pro-vitamin A molecule, it is a precursor of vitamin A. Astaxanthin is not a precursor of any vitamin because there are other carotenoids which are also precursors of vitamin A, but astaxanthin is a carotenoid, but it is not related with vitamin A activity, but it has been found to be a highly health promoting molecule. the point is that the pigments that we are producing from the microbes, on one hand they act as color importing substances in our food matrices, and on the other hand these have health promoting properties. So then we have how it is basically linked with the theme of this thing, that we have been trying and we have been now using agricultural wastes as the substrate for growth of these microorganisms. So these need what are the requirements of these microorganisms for proper growth? They need a carbon source, they need a nitrogen source, they need minerals and vitamins. So obviously if we have extracted something from the fruit, say we have apple poo mix, we have extracted how it got generated, because we had a whole apple fruit, we extract the juice out of it, that is the liquid part of it, and then we have the solid material that was in the form of the insoluble fibers, the peel and other things. But it also contains the substances that are present in the juice, that is, it contains some sugars that may have remained in it. We have not extracted every sugar molecule. It contains these polyphenolic substances that were present in the peel. It contains the minerals and vitamins that were present in the peel or the apple itself, a part of it is contained. So we try to exploit this nutrient basket that is present within these wastes. So the beauty of these microbes is that so the beauty of these microorganisms that we are exploiting for the production of colors is that by certain modifications we can use agro and food industry wastes as a substrate for their growth. And when we are growing these microorganisms, there are two methods of growing these microorganisms. Either we go for solid state fermentation, that is, there is a solid form that is inoculated with the particular microorganism, and then it is kept under given conditions in terms of temperature variability, and then we have to extract the cells from the solid mass and then extract the color from these cells. Or we may have submerged fermentations, wherein we use liquid media. We have monaster, we have Hafia, we have Sarcina, we have Acromobacter. So these produce different pigments, and besides being colorants, these also produce properties like being anti-cancerous, immunosuppressive, antibiotic, anti-proliferative, and positive degradability. So this is an important aspect, that biodegradability. So when we are discussing that natural pigments, these have a tendency that these are not stable during processing or during storage. So that may have a negative impact when it comes to our food supply chain management. In one aspect that we have to take care that our food remains acceptable in terms of color during the storage and after processing. But this has a very positive notion in it also, that being biodegradable in nature means if we consume a lot of colorants of this kind into our body, these will somehow get degraded in our body and will not get accumulated. So that way these are safe from the health point of view. From the technological point of view, this can come as a negative attribute, but from the health point of view, safety point of view, we can reckon this as a positive attribute. So these are the important tools that we are using for production of enzymes, antibiotics, acids, pigments as well. So these are the production technologies. We have solid state fermentation and we have submerged fermentation. So you can see how we can process the wastes over here. We have water waste. We have, for example, this is a brewer's fat green. Brewer's industry waste. We have processed it. We have converted this into a broth. Then we have inoculated monastics onto it. It has been developed this color treatment. It's not done by us. This has been developed, and then we have subjected it to downstream processing, that is extraction of the pigments, and then the colorant has been extracted and converted into a powder. This is a simple process of developing colorants from wastes. So these are some of the microbial colorant pigments. For example, Acromobacter produces a creamy pigment, Bacillus produces a brown pigment, Brevibacterium produces orange and yellow, Corynebacterium produces greyish to greenish yellow, Pseudomonas produce yellowish pigments, Rhodococcus produce bluish and red pigments, and Streptococcus produce yellow and greenish pigments. Aspergillus produces orange. This is an important, that is Black Acidia trispora, creamy pigment it produces. So these are different shades of pigments that we get from the microorganisms. And then among the yeasts, we have Rhodotorula and we have Trachea. These are important. And among the algae, we have Dunarelia. We have these species that we are producing. Now, coming to the yeast part of it, the yeast that we are using for production of the pigments. These we have basically employed in some other applications also. For example, when we go for wastewater treatment. In wastewater treatment, what we are trying to do? We are trying to reduce the carbon content of the water so that it will not support growth once it is once it is sent to the water bodies. So what we have to do? We have to remove the organic content, carbon content from the wastewater. For example, when we generate wastewater from food industries, that is used for cleaning purposes, that is used for cleaning other equipment for the floors. There is possibility that a lot of organic matter that gets added to the water. So it is carbon content increases. And once we are sending this wastewater from industries into the water bodies, we have to subject it to the water treatment. So you have basically seen effluent treatment plants, these are established in the food industry. So one of the main processes that occurs there is removal of this carbon content. And here the algae that has come to our rescue. We are adding algae to it, and algae has a basically role that it will utilize the nutrients, be it nitrogen or carbon. It will act, this will act as a nutrient or substrate for that. It will utilize it for its purpose, and then we can treat the wastewater at the same time because we get rid of the nutrients, and then we can harvest those algae for color production. Okay. We grow algae on wastewaters, algae of the type which are pigment producing. So they have a tendency that they will use the wastewater as their substrate because it is rich in nutrients. It has come from the food industry. It contains nitrogen substances, contains high carbon. It will use it as a carbon and nitrogen source. As I already told you, it has these requirements. So what will happen? It will utilize it. So what will happen to water? It is water. It is carbon content, nitrogen content, nutrient content will be reduced. And that is what we need once we send the wastewater into the water body. It should be free from the nutrients. And at the same time, we can retain these algae being pigment producing, and we can exploit those algae for color production. Okay. So this is Monascus superculus FNJ. It is also called red yeast rice. It produces pigments mainly of the type red, orange and yellowish, and it is suitable for solid state fermentation. This is another orange red pigment. This is produced by red pigment is produced by Rhodotorula species. Here apple pomace have base from apple industry, which is cheap, rich in sugar and minerals. It is used for production of the pigments. The production of Rhodotorula bulletinus by batch fermentation in a rotary shaker has resulted in good yield of carotenoids. This species grows well at 35 degrees centigrade, and it takes around 5 days after centrifugation and washing with the distilled water, we obtain these cells, and the optimum conditions for this are, say we should have a temperature of 30 degrees centigrade, and it takes around 5 days. This is the pigment of our interest. Basically, one of our PhD students is working on this pigment. That is Thakia rhodozyma from yeast, but we are using some other species, again a yeast species. It ferments glucose and other sugars to produce pigment astaxanthin. The pigment is produced in shaking flask at 21 degrees centigrade, and an increase in glucose concentration in the medium gives higher yield of the pigment. Then we have one more pigment, that is beta-carotene. It's a pro-vitamin pigment. It also acts as antioxidant and has properties against several diseases, particularly cancer. And the organism that produces is black HDR trispora, mucor, and hypomycin. And how it is done? That we inoculate the medium, and we incubate it for 8 days under aerobic conditions, and we also keep it under continuous shaking. Then we obviously have to remove the cells and extract the color. So you can here see the waste can be used for this. From 62 kg of corn seed liquor, 38 kg of glucose are produced, and then we can obtain 90 grams of beta-carotene in the end. Then lycopene production. Obviously, this is a pigment from tomato, naturally present in tomato, but this is also produced by microorganisms. Fusarium is the species that is a genetically modified fungus that was first used by Jones to manufacture this under the lab conditions. Then we have one more pigment, that is prodigiosin. It has several pharmacological properties like it is used in several pharmacological applications, and it is produced by Vibrio species. Then we have violacine. It is produced from Prometobacterium, and it also, besides being a pigment, it has several health promoting properties like it is antitumoral, it is antiprotozoan, anti-cancerous, antibacterial, and antioxidant properties. Now, one important thing is that we have basically, now our story is like that. We have a project on this production of biocolors and bioflavors. Production of different kinds of biocolors and bioflavors funded by ICMR. And then one PhD student is working on production of astaxanthin. So these are the two works that are going on in our lab related to this topic. And what we are trying to do over here is that we have in our objectives mentioned that we will be using agro-food wastes for the production of colors. So initially this year we have targeted apple pomace. We have targeted apple pomace as a substrate for the production of these pigments. And what is our objective? Our objective of that research is that we are trying to optimize the production conditions, the growth conditions for the microbial organism, for the microbes that are producing color, so that maximum yield is obtained. So our primary objective of the research, both from the project and from the student work, is that we are optimizing the growth conditions for a particular microorganism for the production of the pigment. So what we have done, we have procured the given strains for production of astaxanthin, and we have procured one that is serratia or red color pigment. We have procured this from the labs outside India. We have procured pure cultures. These are basically how we obtain it. We obtain these cultures as freeze-dried cultures, which are highly stable, and during high temperatures, during storage and other things, these are not degraded because these are stabilized cultures. We have bought these from IMTech and some other national level institutes. We provide these strains against some cost. And then the first step is that we have to revive these cultures because these are not growing there, these are freeze-dried. We have to revive these cultures. How we revive? We give them the best media for their growth. We inoculate the growing media. For example, there is yeast malt broth that is for the these yeast species, if these are. And if we have bacterial species, then we have nutrient agar. So we have basically procured some species of the bacterial type and some of the yeast type. And while working with the yeasts, these are a bit difficult to grow as compared to the bacteria. Basically, their normal growth period ranges from 5 to 7 days. So it takes longer period. Whereas in case of bacteria, the growth period is only 24 hours. At the max, we have to keep it for 48 hours. So that is one thing, that we basically select the inoculum, the microorganism, based on the theoretical knowledge. And then the second part of it is, we have to optimize the growing conditions. What are the growing conditions? There are two factors that basically decide microbial growth: intrinsic factors and extrinsic factors. Intrinsic factors, intrinsic factors are those factors which are related to the food. These include water activity of food. These include nutritional composition of the food. These include pH of the food. These include electric conductivity of the food. These include presence of inhibitors in the food. These include presence of promoters in the food. Then we have extrinsic factors. And important extrinsic factors are gaseous composition around the media, temperature during the growth, and relative humidity. These are the main extrinsic factors. So obviously, if we are optimizing a process, we have to look into both of these factors, because if we will keep killing off the foods, and all those preservation techniques, these are targeted against the microorganisms. And when we study growth curve and factors affecting the growth, we study with this notion that how we can target the microorganisms once we have the knowledge of their growth factors. Now, that is not the only aspect. You are also knowing that we have several beneficial applications of microorganisms, and one of those beneficial applications is that we produce several pigments, we produce several flavoring compounds, we produce minerals from these, we produce single cell proteins from these. So, besides that, these microorganisms are also involved in production of fermented foods. We have n number of traditional or industrial fermented foods. We have all types of curds, we have all types of alcoholic beverages, which all depend on the microbial growth. So now, in this aspect, we have to see that how we can promote the microbial growth. So once we are optimizing the conditions, we have to take care that all the factors which beneficially affect the microbial growth, they are taken care of. So that is, the first is temperature. Temperature should be optimum, and depending upon the microorganism, we can have cyclophiles, mesophiles, and thermophiles. But mostly microbes are mesophiles, intermediate. But within that range of mesophiles, each and every organism has a particular temperature of its own. So it can be a mesophile, it can grow at an intermediate temperature, but every microorganism has a best temperature that may be 2-3 degrees different from the other organisms. So these 2-3 degrees will matter a lot when it comes to microbial growth, isn't it? So we have to take care of the temperature. It can range in certain cases 25, it can be about 35 in some species. Then another is pH. If we have bacterial cultures there, we have to maintain near neutral pH. And if we have fungal species, particularly yeast type, we have to maintain a low pH in the acidic range. So like Rhododendron, it is 4 to 4.5. This is the best pH for them. Then we have carbon source. It should be there, and again we have to just check that if the sugar content in the waste is sufficient to support the growth. If the sugar content, the fermentable part of it may be a resource of carbon, but a part of it may not be digestible by the microorganism, fermentable by the microorganisms. So in that case, once we are dealing with the waste, that's very important, that we are enriching the waste, we are analyzing the waste first for it is the nutritional composition, then we are suitably enriching the waste with a deficient nutrient. There may be deficiency of the carbon source, obviously we may have to add maltose into it. There may be deficiency of nitrogen, we may have to add yeast extract to it. So depending upon the composition of the waste, we have to modify them. So we cannot straight away go procure the waste and go to the lab and put the microorganism into it. We have to take care of the requirements of the microorganism, and accordingly we may have to enrich the media. Then types of the fermenters, again this we have done, that if we have a simple fermenter like an incubator type, then the growth is very slow. And if we have a shaking type of incubator, because in case of the static type of incubators, the immediate microenvironment gets saturated. So the growth gets slowed down. There are nutrients in the bottle, but in the spots where there is large microbial growth, there is deficiency of the nutrients. So if we keep shaking, if we keep our setup shaking, these nutrients will be transferred from this spot to that spot, and that will result in that there will remain no unutilized nutrients within our media. So that is basically the type of fermenter that is effective. And again, solid state and that versus that is important. So these are some of the applications that these are used in cosmetics, these are used in today's tools, these are used in textiles, these are used in medicine. Let's cut it short. These are the images from our lab. We have used this Serratia marcescens. This is the strain straight away from the lab that we have procured from Intech. And this is the first plating on the nutrient agar to revive these. But the real process is that it is to be done in the submerged fermentation. So we have used the cloth, and then we have centrifuged it so that these cells remain in the tubes and we remove the supernatant, and then we have preserved these cells in the 15% glycerol before we will subject it to basically disruption and extraction of the pigments. And this is another experiment, that is astaxanthin production by xanthophyllomyces. Hafnia is there and there is xanthophyllomyces. This is the slant that we have procured from outside, and this is the plating on PDA, and then we have cultured it in the yeast malt broth, and after centrifugation these are the cells. This is not pigment, these are simple cells, and afterwards we have extracted the pigment from it, and that has been that is to be then used further. So this is the thing that we are doing. Now one important aspect that is there, that is we have to go for optimization of these conditions. What is the temperature? What is the pH of the media? What is the nutrient composition? So that is to be seen. That is what we are experimenting over here. Number one. Number two, with all the biological kind of pigments, be it from the microbes or be it from the wastes, as I earlier said, extracted from the wastes, there is stability issue. So in order to stability at two stages, during the processing and during the storage. So in order to make these pigments stable, we go for their encapsulation. So that is another experiment, that is encapsulation of these pigments, and we have to be very careful while we are going for encapsulation. Encapsulation should be such that once it is added into the food, its color should not be masked by the wall material. Number one, and number two, so encapsulation what it does, it helps its preservation during processing. It will not basically get degraded during heating. And then during storage, once it is exposed to light, once it is exposed to oxygen, it gets preserved in encapsulation. But one more important thing that is associated with encapsulation is that, for example, if we want to use keratinide type of pigments, keratinides are fat soluble. If we are using keratinide type of pigments and we are using it in aqueous system, so there is dispersibility issue, there is solubility issue. And our wall material, once we encapsulate it, it can resolve this issue. That insoluble pigments, these can be, by choosing a proper wall material, we can solubilize it in a matrix wherein these are insoluble. That's an important thing of encapsulation. And then in our experiment, we are experimenting that we will be trying to make apply this into ice creams or coloring of ice creams we are initially trying, so that the damage by heat initially is basically avoided. Later on, we can think of using it in some beverages or juices which are thermally processes. And one more thing is that one of the applications that I just forgot here is that these colorants are hugely, largely used in fish feed. These particularly astaxanthin pigments. These are used for coloring of fish feed. So what it does is that once it gets into the nutrition of the fish, we have those beautifully colored fish species. You have seen colored fish species, and this astaxanthin has several other benefits. But its main source is one of the main sources is also animal sources, that is fish species contain this astaxanthin. And we can make fish species very rich in astaxanthin if we develop a feed which has been incorporated with the astaxanthin pigment, and it moves into their food system and it comes into their bodies. This is how we make omega-3 eggs. Omega-3 eggs are made by feeding the poultry birds with omega-3 rich foods. In others, incorporated, for example, we have omega-3 bread, we have omega-3 jam. They will be incorporated part of omega-3 into the recipe of it. But when it comes to omega-3 eggs are here, it is to be incorporated with the feed of the poultry birds. In a similar fashion, in fish species, we have several colored fish species, and it is not just for decoration purpose. We can get very good health benefits out of the consumption of these fish species. These are enriched. Their feed is enriched with these pigments. This is one of the applications of that. Okay. So I think I will finish it over here. So any questions are there?
Sir, बहुत सारे requirements like आपने कहा fruits and vegetables select from the carrots from pumpkin या encapsulation. Encapsulation करने के बाद भी they are not stable. फिर भी वो degrade होते हैं. तो हमारे पास क्या खाली एक option है कि हमें fruits and vegetables से pigments में replace करना है.
मैंने पहले बताया इसमें दो चीजें हैं. सबसे बड़ी चीज जो है इंस्टेड ऑफ स्टेबिलिटी ज्यादा इशू है यील्ड का. सो we can say in terms of say celosia may be a good source as compared to your pumpkin peel or beetroot, because celosia is very much abundant in these pigments. So that is not the stability issues may come in these pigments also. Say it is lycopene, whether it is from tomato peel or it is from the microbial organism. Stability issues are there in both the cases. But the thing is, if we have a safe pigment, what is the solution? We can add it in, say double the quantity than what is required, so that during processing, a part of it is degraded, a part of it will remain stable to make it acceptable. So that can be done in this. So once we have to add more and more colorants. So for that we need more and more of the colorants. And when it comes to yield, in terms of yield, say carrots is 1 kg of carrots. I will just tell you the exact data. Yeah, if say carrots take around 100 days for the life cycle, and if we will use this for beta carotene production, 70 mgs per kg will be produced, mgs. Whereas if we will use microbial production, the organism will take 5 days, considering the say worst case scenario. 5 days से कम भी लगेगा. It will take 5 days, and during 5 days it will produce 250 mg per kg of the biomass. So that is the thing. So that is kg of carrot. When we extract it, 1 kg of carrot, it will take 100 days for you to grow that carrot or celosia or some other crop, and then we will extract it. While how this is a better alternative that within 3 days, within 2 days, within 5 days, at the max 7 to 8 days. In case of yeast, the growing cycle is more. It takes 7-8 days at least. You can get twice, thrice the quantity per kg basis. So that is the basic beauty with this, that even if these pigments are degraded, we have sufficient pigments with us. We can add double the quantities. These are very degradable, and if there is no safety issue with these molecules, so there will be no restriction when it comes to dose. So that will be the basically alternative how these can be helpful. When it comes to stability, these are at the same level when it comes to the pigments from the fruit and vegetable wastes. Then one more thing is that in fruits and vegetable wastes during extraction processes there may be further degradation. That will be clear. Yes, yes. देखिए स्टेबिलिटी में बहुत सारे इश्यूज हैं. लेट मी कोट माय एग्जांपल्स जो मैंने आपने खुद से देखा. कैरोटीनॉइड्स जो होते हैं ना, दीज आर significantly heat stable. Significant to a very significant level, these are heat stable. For example, I did canning of apricots. Canning means boiling कम से कम है ना 100 plus. So there was loss of texture, but there was no loss of color. No loss of color means visibility. ऐसे chemically तो हुआ होगा pigment loss, but visibly apricots were as yellow as they were on the tree. So there are stability issues, but then in case of anthocyanin, there is one more thing. Their coloration is pH dependent. In one pH they will show one shade, in another pH they will not show any shade at all, in some other pH they will show some other. So when it comes to stability issues, there are stability issues that is there, but it is not that कि लगाते खत्म हो गया. Anthocyanins के साथ मसला है. So that is why anthocyanins are red in color. So if you have some reddish color pigment, that is of the carotenoid nature. Some modification of that we can do that. And one more thing that I forgot basically, that is the research into this microbial production of pigments is that we have to develop very efficiency trains. By way of mutation, we can say what is this process all about. मैंने आपको पढ़ाया भी होगा that the biosynthetic pathway for production of pigments, be it anthocyanins or be it carotenoids, isn't it? There are pathways. There is mevalonic acid pathway. There is shikimic acid pathway. So there is a precursor molecule, acetylco enzyme. Then it's changed. Then it is changed. Then with every change we require some enzyme. Obviously, when this enzyme setup is present in plants, and obviously this has to be present in the microbe so that it produces the same pigment. Beta carotene बनने के लिए जो carrot को लगेगा setup वो microbe में भी होना चाहिए. Now what we do, we make some very efficient strains which have high levels of all those enzymes, so that they can produce higher yields and higher levels. So that can be done. But when it comes to stability, stability में याद रखिएगा, it is not that कि लगाते खत्म हो गया. There is issue with the anthocyanins, but there is not that much issue with the carotenoids. But when it comes to stability issues, be it from the plant source or microbial source, they are at the same level. There is no difference. But we can produce more, we can handle it more efficiently because everything is in our control. There you have to destroy the cells, those plant cells, they have a different setup, so that can be very good. And these are commercially done, this is not good. मतलब industrial scale पे this biocolor production, it is around $1.5 billion business. So it is not that we are just trying, we are just replicating the things in our lab. Otherwise, it has been done and it has been commercialized. So now our part may be that we are using our own waste, the apple pomace and other things. That may be an additional. Otherwise, it is always it is a commercial kind of activity. It is not a sort of research. It is not a research. Ji, any other? Okay, thank you.
Thank you so much, sir, for such an insightful lecture. I learned a lot of points from it. So it was really a very great lecture. Thank you so much. Now I request sir to receive a moment of